This is an overview of an article entitled Improving Interpretation of Cardiac Phenotypes and Enhancing Discovery with Expanded Knowledge in the Gene Ontology, published in the February 2018 issue of Circulation, Genomic and Precision Medicine. High-throughput genome-scale methodologies, such as genome-wide association studies, exome sequencing studies, transcriptomics, and proteomics, are increasingly being used to study cardiovascular traits and diseases. Interpretation of these studies relies on high-quality descriptions of cellular and physiological roles of genes and proteins. To support data interpretation, the Gene Ontology, or GO, consortium provides a freely available, structured, controlled vocabulary that enables association of defined terms, describing cellular roles and locations with an RNA transcript protein. The GO resource provides a computer-interpretable summary of the results of many independent experiments. GO data has been incorporated into over 50 functional analysis tools and is routinely used to analyze large data sets. In order to improve the cardiac GO resource, a coordinated working group of GO editors, GO biocurators, and field experts expanded the cardiac physiology domain by adding 197 new cardiac-relevant GO terms. This expansion allows for the description of the roles of genes and proteins in specific tissues of the heart, in specific cardiac cell types, and in the whole organ. The expansion includes terms that reference specific anatomic structures, but also includes generic terms that can be applied to any organism with a cardiac structure. Thus, the GO resource is not limited to genes and proteins in humans and other mammals. Examples that employ the new GO terms include adrenergic receptor signaling pathway involved in cardiac muscle relaxation, AV node cell to bundle of His cell communication, and regulation of the force of heart contraction by cardiac conduction. This schematic shows a portion of the ontology describing AV node cell action potential and the regulation of ventricular cardiac muscle cell action potential. Here you can see how different terms relate to each other. In some cases, when two terms are connected, there's a parent-child relationship in that one is a part of the other. In other cases, the relationship is one of regulation, where one regulates the other. These relationships allow for highly specific descriptions of biological roles. This table shows a subset of the GO annotations for the human ACE2 protein using the expanded GO terms. Each of these annotations is based on experimental evidence. In total, there are 61 annotations for the human ACE2 protein. Here's an example of how the expanded cardiac GO resource can be used to nominate causal genes in genome-wide association study, or GWAS, loci. Shown here are 3 GWAS loci with DNA variants associated with the electrocardiographic QT interval, which reflects the duration of cardiac repolarization and, when abnormal, can signify increased risk of sudden cardiac death. Each locus harbors multiple genes, any one of which could be a causal gene that contributes to the QT interval. Upon applying GO annotations, one sees that in each locus there's one gene that stands out with respect to GO terms that are plausibly related to cardiac electrophysiology. Although the GO annotations don't prove that the genes are causal, they can serve to prioritize those genes for experimental study. Here's an instructive example that shows the utility of the expanded cardiac GO resource in comparison to the previously available resource. Transcriptomic data was used to identify the 200 genes most differentially expressed between adult human ventricular cardiomyocytes, and undifferentiated human embryonic stem cells. By tabulating GO terms associated with any of the 200 genes, one can identify biological processes that distinguish the two cell types and, presumably, define the cardiomyocytes. Having new GO terms available, the expanded resource, the one designated with the year 2016, is able to ascribe mitochondrial organization, cardiac conduction, regulation of cardiac conduction, and regulation of the force of heart conduction as characteristics of the cardiomyocytes, whereas this was not possible with the old resource, the one designated with the year 2011. In conclusion, the expanded cardiac GO resource supports a more informative interpretation of genome-scale data. This can help, for example, to identify candidate causal genes in genetic association studies. The effort to improve the cardiac GO resource provides a structured framework to improve the computational representation of biological knowledge in all fields.